So we know that fertilization, no, right? What once the pollen tube reaches the ovary now and penetrates the ovules and go through the micropyle, right, to go into the ovules, right? Fertilization occurs, right? So this process involves the fusion of the male and female gametes. That is what fertilization is. That's the essence of it, resulting in the formation of a zygote and initiating embryo development. In most angiosperms, double fertilization occurs, and a unique feature distinguishing them from other plants. Alright? So let's look at this now. Double fertilization. Why is it called double fertilization? Anybody?
Because both of the gametes fertilize the mature ovule.
Okay. Mhm.
One of the gametes will fertilize the, so I think it's the generative nucleus that will fertilize the egg,
Mhm.
and the other one will fertilize the,
The
I don't
endosperm
remember.
nucleus.
Okay. So it's called double fertilization because firstly we have the general fertilization where one of the sperm cells will actually combine with the ova, right? And actually, which is the egg cell, and cause the creation of the zygote, while the other sperm nuclei will actually now go and combine with the two polar nuclei to create the endosperm nucleus. All right, lovely. So in double fertilization, one sperm cell fertilizes the egg, forming the zygote, which is diploid, and then the other now, right, will actually go and well that will develop into the embryo, and the other one now will create a triploid cell, right? Because what we have in the sperm cell has only one nuclei. So when it now combines with two other polar nuclei at the center of the ovule, it will become a triploid structure, right? And this is what we call the primary endosperm nucleus. Right, so this triploid nucleus then divides to form the endosperm itself, a nutrient-rich tissue that nurtures the developing embryo. The endosperm is crucial, right, for the embryo's development because it provides the nutrients, right, such as proteins, oils, and starch that the embryo would need for embryonic development. All right. So the significance of the ploidy, we know the significance of ploidy, right? So we know what when something is haploid, diploid, polyploid, right? It just means that it has different amounts of the genetic information, the different sets of chromosomes, right? So in zygote, right, the zygote will result right, from fertilization as a diploid cell containing two sets of chromosomes, right? So one for each parent. We know how that works. Okay? So in contrast, the endosperm formed will have a fusion of the sperm cell and the two polar nuclei, giving a triploid cell containing three sets of chromosomes. The difference in ploidy levels between the embryo is 2n and endosperm 3n ensures proper development and nutrient supply to support the growing embryo. Alright? So basically the triploid cell creates the the triploid cell is generally inert, right? It will actually just be filled with nutrients that the diploid cell can now leach off of, basically. Alright? So, first thing, fusion with the egg cell, right? Second thing, fusion with polar nuclei. So, when we have the first fusion with the egg cell, create our zygote, which is diploid. When we have our second fusion, we're gonna have that fusion with the polar nuclei to create our triploid cell, which would produce our endosperm nucleus, right? And the endosperm nucleus now, that cell will now actually develop into our endosperm. Okay, so this is what's happening now. This is most definitely what we have happening. All right, so we have everything that I just discussed on one slide. What we're looking at here now is pollination. Pollination, the pollen grains lead from an anther towards a stigma, right? And what's happening here now, that is now pollination. We have germination occurring where the pollen tube begins. The pollen tube will continue to grow right? Until around 5 here, that you see it enters now, right? The micropyle, right? One of the synergids would actually degenerate, allowing, right, for the egg cell to now be fertilized into the zygote, and then the other nuclei will pass through, right, to bond with the two polar nuclei to create a endosperm nucleus, right? As it develops now, as we develop now, the antipodal cells and the synergids will disintegrate, they will degenerate, right? And then we have the creation of that endosperm with the embryo, right? So the endosperm will now surround the embryo, right? And then we have further embryonic development. We're gonna talk about the further embryonic development now, all right? So let's look at what's happening here, right? One, initially the pollen tube contains two haploid cells, the generative cell, right, and the tube cell, right? Then the generative cell divides mitotically, right, so through mitosis producing two haploid sperm cells, and then the sperm cells enter the cytoplasm, right, of a synagid, right? So remember what I said, we enter the cytoplasm of a synagid, the synagid will disintegrate, right? Then the other, then one now will actually continue to actually inhabit, right, the egg cell, right, and then their genetic material will mix, creating our zygote. And then lastly now, the last sperm cell will actually now fuse itself with the two polar nuclei, creating our triploid endosperm cell, right, or endosperm nuclei. So that's what we have there. This is what we call double fertilization, right? So when we enter now, we're gonna have the degeneration of a synagid, right? The egg cell will now be fertilized by a sperm cell to create a zygote, and then now the polar nuclei will now form with the other sperm cell to create our endosperm nuclei, and that is it. Any questions? Okay. What happens after fertilization? After fertilization, right? We know that fertilization does. We know, right? So following double fertilization, the zygote undergoes rapid cell division and differentiation, leading to the formation of the embryo. The embryo typically consists of several distinct regions, including the embryonic axis, right? Which will give rise to the plumule and the radicle. And then what we have now is one or more cotyledons. So if we have two cotyledons, if it's dicotyledonous, we're going to, that is really shown in plants due to its complex web-like vein system on the leaf. And if it's monocotyledonous, we tend to see parallel running lines around its leaf structure. You guys know monocot versus dicot leaves. You have done that in lower school, right? Much less C-set biology, right? So we've done that before. So we're gonna have two different cotyledons, or one cotyledon emerging, right? And our embryonic axis now will give rise to our plumule and our radicle. Which one is the root system? Plumule or radicle? Which one is the root? Is the derivative of the root system? Is it the plumule or the radicle? What is the plumule or radicle? Which one is root and which one is shoot? Go ahead.
The radical.
The radical is,
Radical is the root.
all right, and the plumule is the shoot. All right. So this is what we have here.
Shoot.
Lovely, right? So this shows the stage of the embryo development in the ovule, right, of shepherd's purse, or what we call capsula bursa, right? So this is just looking at the capsula bursa and what happens after its double fertilization, right? So we're gonna have the creation of the basal cell, right? And we'll have a suspensory ligament that attaches itself to a proembryo, right? And then the proembryo now will develop into an embryo, and the embryo itself will now grow cotyledons, right? And now it will actually grow over into our endosperm, right? So the embryonic axis produces two cotyledons. In this case, it's two cotyledons, because if you look at Capsula bursa, it is a dicotyledonous plant, right? and now it will grow to now grow into the endosperm. And you know what's happening to endosperm here, right? As you see, we go through the stages, the endosperm is being consumed because we consume the nutrients of the endosperm, right? And then now we have this structure where we have a short meristem here at the junction of the two cotyledons, right, to the embryonic axis, right? And then we're gonna have a seed coat. What is the name? What's the other name for a seed coat? What's the other name for a seed coat? You guys should know what that is. We don't remember the other name for the seed coat. We should. I'm gonna allow you guys to figure out the other name for the seed coat. Alright. So we're gonna have a seed coat being formed, right? So the ovule itself, right? The outer layer of the ovule will actually harden to create the seed coat, right? Our top meristem there will be the will be what where we have the plumule coming from, and then we have our root meristem right down here, which would be our radicle coming from, right? Where our radicle will be coming from, right? So that is how we have the embryonic development within the plant there. So this is another way of looking at it, right? We have a ground and we have a procambium, right? We have the hypocotyle, right? These are many different structures that we will see, right? Many different structures that are named differently depending on which source you go to. So in the case here where we have our short meristem and it will grow from what we call the procambium, right? Well, the procambium is actually in the middle, right? It's around here, right? And then it will develop, right, into what we call a hypocotyle. Well, hypocotyl, right? So we have that there. We have the shoot apical meristem and we have the root apical meristem and we have the two cotyledons here. But we went through the process, right? So we have our proembryo connected to our suspensor ligament connected to our basal cell, right? And then it is our proembryo now which will create our embryo, right? Which will divide, right, on along its embryonic axis to produce two cotyledons, right? And divide along a junction where we call the procambium, right? And then now we will have the development of a large thick hypocotyle, right? And our shoot meristem will come from, our shoot meristem is between the two cotyledons there, right? At what was once called our procambium. And then at the bottom now close to our basal cell, the suspensory ligament there, we're gonna have our root meristem being grown from. Alright? So that's what we have there for the embryonic development within angiosperms. Okay? So this is another look at it. Alright? So when it's fully grown, then that's what we're gonna call a seed. Okay? So that's why this says developmental stages of the seed. So it's the same way when you look at a pea plant in C-sec and the little divots. If you look at a red bean or a kidney bean or a red pea, right? Depending on which Caribbean country you originate from or which country overall, right? You're gonna have a divot in the in the pea, and you know that divot is a hypocotyle, right? And then the overall red coat on the pea, that is our ovule layer that has hardened to become our seed coat. And then when you cut into it now, you can see the separation of the cotyledon, right? You should be able to see these things when you actually pick up a seed and look at it, inspect it, right? It is the production, right, or it is the product, the finished product of this entire embryonic development, right? So seed maturation now, as the embryo and endosperm develops, the surrounding ovary walls begins to mature and undergo changes. This process involves the transformation of the ovary into a fruit, which encloses and protects the developing seeds, right? The fruit undergoes various physiological and structural modifications, such as change in color, texture, and chemical composition to attract dispersal agents, right? So it's usually going to have the creation of different types of different amounts of sucrose and fructose, right? So it can attract animals or animals that will consume the fruit, and then through egestion, seed dispersal will occur. All right. So fruit development, we know that our fruit will go through different areas of development, right? We're going to the creation of our different layers, right? So this is not too much, right, of something that you guys would actually go through. But understanding how the fruit is developed is something really simple, right? And we know we're going to end up back to what we saw in C-sec biology, where we're gonna have the creation of the parts of the fruit, right? So our endocarp is what we call our seed coat. I want you guys to get that word, the endocarp, right? And then the mesocarp itself, right, is what we're gonna call our fleshy part that you eat. You eat the mesocarp of the developed ovary, right? And then our epicarp will be the skin of the seed that you tear off, right? So our endocarp can form the seed coat, right? The seed layer, right? And then here you have the embryo inside and the endosperm itself, right? So the seed itself. Okay? So those are general parts that we get from all of this development. And that is the end of sexual reproduction in plants. Alright? So that is it.